Okay, so this is our first case study for looking at P53. So we've already looked at mutations across the cancer genome atlas, the 32 different cancer types, and we're sitting here now at the cancer type summary plot. That's how we finish the core part of the practical. So now it's time to investigate a few of the different plots. Now, immediately from this plot, I can see that most of the mutations in these different cancer cohorts across the bottom are in green here, these green bars. And that means that these are mutations, standard mutations. However, we can see here in the sarcoma dataset that whilst lots of them are green, we've also got these blues, so that's deep deletions, so that's loss of both copies of TP53. And you've also got some other structural variants as well. So that's where there are bigger changes. That could be amplifications, it could be a shallow deletion, so losing only one copy on average, or it could be something else that's going on. So what we're going to do now is we're going to explore this sarcoma dataset. So we're going to query this study for TP53. So just like in the core part of the practical, you can use the plots here to look at the oncoprint. You can see 47%, so that's higher than the average. So there's lots of mutations here, lots of deep deletions. But if you're looking at the different types of mutations, you can see that just like in our work before, we've got this big hit of mutations happening across the DNA interacting domain, including the mutations that we were looking at in our practical. Okay. Now, even though these are quite common, if we actually go in and look at survival, you can see there's no difference. Now that's strange because they're really, really common, these mutations. They're having the same impact as we saw across the whole TCGA cohort, but with TCGA as a whole, there was a significant difference between these two groups. So what's going on here? Well, the answer actually comes down to the fact that P53 doesn't act alone. So in biology, proteins act together in pathways, they don't just act on their own. So we're going to look at this pathways tab, which we haven't used before. Because we've just searched one gene, TP53, we've only got a limited number of pathways coming up. If we'd search for lots of different genes, lots of different pathways would appear. The numbers, the percentages underneath each of these genes represents how many times this gene is mutated in the cohort. So you can see here under P53, in sarcoma it's 46.7%, but you can see here as well that these two genes nearby to TP53, CDKN2A and MDM2, they're also mutated quite a lot as well. Now what these arrows mean, so an arrow means, so if an arrow goes from one gene to another, it means that in this case, RPS6KA3, nice catchy name, is promoting the activity of P53, whereas an arrow that ends in a single black bar, that means that it's inhibiting the effect of P53. So that's interesting. MDM2 and CDKN2A both inhibit P53. Now, if the cancer is trying to inhibit P53 overall, why would there be mutations in these genes? It doesn't really make sense. So what's actually happening here? Let's find out. So we go up to this little edit, genes at the top, and we physically type in MDM2 and CDKN2A. Okay, so all symbols are valid. We can submit this query. So now we're not just focusing on P53, we're focusing on everything. So you can see here, these are now highlighted dark black borders to show that we've selected everything. We can head back to our oncoprint, our usual landing spot. Now you can see here at the top corner, 74% of samples now have a mutation. So P53, it was 46 or 47%, whereas now it's 74%. That means that these mutations are not all happening in the same people. And you can see that here in the oncoprint. So most people, well, almost half of people have a mutation in P53. CDKN2A, 15%, they're kind of dotted about. And MDM2, 21%. But interestingly, there is this disconnect between P53 and MDM2 mutations. So if you have a mutation in P53, you probably don't have a mutation in MDM2. It's not as clear with CDKN2A, but there's still this big block at the end, and very rarely do CDKN2A mutations occur at the same time as MDM2. Now this is a phenomenon called mutual exclusivity, and you can see here there's a tab now that's appeared at the top. We can click on this. It shows the different comparisons, and you can see that it's significant that TP53 and MDM2 mutations do not occur together, and similarly, MDM2, CDKN2A do not occur together. For P53 and CDKN2A, that's not a significant difference. So why does this matter? Well, because these genes are acting in a pathway, the tumours only need to break that pathway or modify that pathway once. So there's no real survival. Remember, these are evolving tumours. There's no real survival benefit to the tumours to mutate all the different parts of the pathway. It only needs to hit that pathway once. So now if we switch to the survival plots as before, now we can see, now that the altered group, the red group, has all 3 genes in it, all 3 types of mutations. You can see that now there is a big significant difference. And so hitting the P53 pathway itself at any point in that pathway is bad for survival. Now we said that MDM2, that gene in particular, stops P53 from working. So why would you mutate that? That doesn't really make sense. Well, actually, if you look at the oncoprint and you zoom in, you can see that the mutations that are happening in MDM2 are all amplifications. So yes, MDM2 stops P53 activity. In these cancers, you're actually increasing the amount of MDM2, so it's more likely that it's able to stop P53 activity. So this is really, really important. Multiple different ways of breaking a pathway. So that's the sarcoma example. Hopefully you'll come back for example number 2.